So today we're going to talk about something we started talking about last time, and that's all about stem cells. And why do we want to talk about stem cells? Is because stem cells obviously they grow just like any other cell, but they also through a process called differentiation, they specialize. Okay, and which means that we have stem cells that can do this. This little blastocyst looks like they told me I could do anything I wanted, and so the cell did. Okay, and stem cells, they're so great that they deserve two memes, because stem cells, they're curing diseases without surgery. Here's to you, stem cells. All right, so have a go at these three quick questions and pause the video here and we'll go through them in 2, 1. All right, let's go through question 1. The crossing over of chromosomes occurs in what stage? Hopefully you recognize that it's in prophase 1. Question 2 is asking specifically about banana plants and how banana plants can reproduce asexually, as shown as this in this diagram. All right, we want to know about this form of vegetative propagation, and as you can see, this little banana plant actually has a mini banana plant attached to it, but you can see underneath the ground, it's all the same plant. Okay, and that type of asexual reproduction is called a rhizome. Okay, and this little thing is called it's really quite easy to remember, but it's called a sucker because it sucks from the mother plant. Okay, and how this type of asexual reproduction works is quite simple. This sucker starts growing, this rhizome grows, okay, and by growing, it's going to produce a genetically identical plant that can be cut off and grown somewhere else. Okay. I don't expect you to know all the very much specifics about that, but if presented with something like this, then this would be a good enough answer. Question 3 is talking about identifying an issue related to the overuse of cloning, which is a form of asexual reproduction, I guess, in agriculture. Okay, and there's lots of different issues. Okay, so lack of genetic diversity is going to be the major issue, but this lack of genetic diversity leads to prone to disease. Okay, it's also prone to genetic diseases. I'm going to say diseases. All right, which sounds a lot like that, but here we mean to outside diseases and pests. Okay, so the difference between these two things is that these genetic diseases might be things like specific features of a ball, for example, which might mean that it's not going to survive as well. Prone to outside diseases, pests might just be that it's prone to a specific virus or a bacteria. Okay. All right, so in the study design, we're still in this area of the study design about cell growth and differentiation, but this time we're going to look at more specifically stem cell differentiation, okay? And we're going to look at how they help in terms of human development. All right, so we want to know, how do we get from A to B? All right, how do we get from this guy, which is a zygote? We know it's a zygote because it's only just one cell. It is fertilized, but it is just one. To this guy, which, if you don't know, that's called a baby. Right? So what we have to do, remember, is we have to go through various stages of growth and differentiation. Okay, so what you can see here is the blastocyst. Okay, and the blastocyst, it's the stage in embryonic development where the cell has about 300, well, the embryo has about 300 cells, all right, and these cells are going to start undergoing the process of differentiation. Okay, and what that means is that we're going to get three different layers. Okay, and we talked about these three different layers. Let me just move myself over here. And these three different layers are the endoderm, oops, sorry, the endoderm, the mesoderm, and the ectoderm, and they're going to become different parts of the new human or the new dog or whatever the animal it is. Okay, um, and that's going to be really, really important. Okay, let's talk further about this process called differentiation. So, stem cells, they're cells that, through differentiation, can become a variety of types of other cells. And the reason they can do this is because they're actually able to turn on and off specific genes. Okay, so as you can see here, we have this original first stem cell, and they obviously, through mitosis, can go and become more different stem cells. But also, stem cells, what's really important is that they can go and become skin cells or muscle cells or red blood cells or neurons, all right? And all of these different kinds of cells are all going to be very important. Okay. Um, two different kinds of cells that you guys have to know about. They're called embryonic and adult stem cells. Don't get confused, okay? Because first thing, embryonic cells, they're undifferentiated or relatively undifferentiated stage of the cells, okay? And they're pretty much just present from the zygote, so during fertilization, to the blastocyst stage, so about 300 cells, okay? Once we're passing this blastocyst stage, we actually then call every stem cell an adult stem cell, okay? And adult stem cells, they're present in very small numbers in a very specific tissue, such as your hair follicles, your bone marrow, your spinal cord, and your germline cells, which are in, as you guys know, your gonads. Okay, so those cells that become the sperm and the egg, they're actually a type of adult stem cell. All right, so let's talk about this differentiation. And we actually have different degrees of differentiation, and that's about what we call cell potency. Okay, there's three different degrees of differentiation that you guys have to know about. The first degree is called totipotent. So I remember toti sounds like totally, all right, and they can then go on to become any different cell in the body. Okay, so they're totally potent. Okay, once those totipotent cells go into the blastocyst stage, it's the blastocyst stage. So this is the zygote. Once they go into the blastocyst stage, they then separate into those three different layers: the endoderm, the mesoderm, and the ectoderm. All right, and those three different layers then go on to becoming different cells. And they go from being what we call pluripotent to being multipotent. Okay, and multipotent it's the lowest level of stem cell differentiation capacity. So that's the lowest potency level, we say. And this is what adult stem cells are. Okay, and these are going to go on to becoming all your normal body cells we're used to. Okay, so what's so important about stem cells? Obviously, taking up the fact that the only reason that we have hands and a liver and brain and eyeballs is because of stem cells. But it's also very important in terms of medicine, okay? Because we can actually use stem cells and different stem cell therapies to actually grow and repair tissue in a very non-invasive process. And that's pretty awesome if you ask me, okay? So different potential uses we can see down here. So there's uses such as healing wounds. That's obviously quite easy, but also there's lots of uses all around our body, from the brain to your intestines, right, to actually fix different disorders and different diseases through this stem cell therapy. But there's three different necessities that stem cells have to have, the specific stem cells we want for us to use them in stem cell therapy. The first one is that stem cells must be able to differentiate into the specific tissue we want to grow. And that's very obvious. You're not going to put a stem cell that's going to grow into bone tissue into your brain, are you? Okay. The second one is that stem cells must have controlled growth. And the reason is because cells that don't have controlled growth, we call them, you would have heard of them before, they're called cancer cells. Okay. So we must be able to, in some kind of way, control the growth of these stem cells. All right. And then last, but certainly not least, is that the recipient's immune system has to actually be compatible with the donor. Okay. So you can't just take stem cells from anyone and throw it in yourself. Okay. It has to be someone that, because of the blood typing and other different factors, there must be a compatibility. And that's what raises actually something that is self stem cell therapy, where if we were able to take stem cells from a baby, well, not a baby, an embryo in a very early stage of development, store those stem cells, later on in life, if that baby grows up just having a specific disorder or disease, there's potential for us to use those stem cells that we have stored to cure that baby's disorders. All right, which, if you ask me, sounds pretty cool. Obviously, it's going to be an issue in terms of genetic disorders, all right? But if it's a wound or something that happened during that person's lifetime, this is really, really interesting technology. But like any process in biology and in science in general, really, there are ethical considerations, okay? And the use of embryonic stem cells initially actually involved the destruction of the embryo, at the blastocyst stage, okay? And this caused a bit of an uproar, okay? People didn't want to pretty much fertilize eggs and pretty much get a person started and then destroy that person, okay? The good thing, though, is that new technology today actually doesn't involve this destruction, but unfortunately, the stigma has stuck, okay? So here you can see Simpsons, okay, very much criticizing stem cell therapies. This lady's serving him stem cell fajitas. All right, and here's South Park, and they're actually criticizing stem cell therapies. If you go, I'm not going to recommend you watch the episodes because I can't recommend you watch South Park's, but hey, if it's on in the background, you should watch it, because this episode of South Park also very much criticizes stem cell therapy, okay? But as much as the technology still actually really does have a long way, the potential healthcare benefit of stem cell therapy really is limitless.